Hello and welcome to this series of lectures on understanding bias in the introduction of variation as an evolutionary cause. In the previous two talks, we reviewed the evidence that internal mutational preferences impart preferences to the process of evolutionary adaptation so that the changes involved in adaptation are enriched for what's mutationally likely. This is interesting for two reasons. First, it establishes a new connection. Just a few years ago in this Nature paper, Houle and colleagues began by saying mutation enables evolution, but the idea that adaptation is also shaped by mutational variation is controversial. But now we know that adaptation is shaped by biases and mutation. The second interesting aspect is that explaining mutation-biased adaptation requires a theory absent from the modern synthesis and classical thinking more generally. When Haldane and Fisher addressed this issue of variational biases, they treated mutation as a mass action pressure within a paradigm in which all the alleles relevant to the outcome of evolution are present initially, so that evolution is just shifting gene frequencies. And they concluded that because mutation rates are small, mutation is a weak pressure, easily overcome by the opposing pressure of selection, important only for neutral characters, factors of negligible advantage or disadvantage, as Haldane says, or in the case of large mutation rates. In this way of thinking shaped 20th century thought about evolutionary causes. In his 2002 book, Gould cites this very argument from Haldane and Fisher to conclude that the smallness of mutation rates sounds the death knell for internalism. That's how evolutionary biologists thought about this issue for 75 years. But to understand mutation-biased adaptation, we have to go outside that framework used by Haldane and Fisher to consider an evolutionary process dependent on events that introduce new alleles. Mutation proposes new things, and these are either accepted or rejected. To understand how this kind of proposal process can influence evolutionary trajectories, think about a mountain climber on a rough, mountainous landscape. A human climber would scout out a path to the peak, but we need to think in terms of a blind process that operates by strictly local rules. So let's imagine a sightless climbing robot or a climber that's operating in complete darkness by a two-step procedure. So in the first step, the proposal step, the climber or the climbing robot is going to reach out with a hand or a foot to sample a different handhold or foothold. And each time this happens, there'll be some chance of a second acceptance or commitment step in which the climber grasps that point of leverage and repositions himself on that basis. If we bias the second step, the acceptance step, such that higher points are more likely to be chosen, this will cause the climber to ascend, to go upward. But we can also imagine imposing a lateral bias on the proposal step. Suppose that our climbing robot has longer or more active limbs on the left side than on the right side. So the climbing robot samples more points of leverage on the left. The joint probability of proposal and acceptance is higher on the left, so the trajectory of the climber will be biased, not just upwards, but to the left. If the landscape is rough, the climber will tend to get stuck on a local peak that's up and to the left. If the landscape is a smooth hemisphere, the climber will spiral upwards and to the left, ascending to the single summit. And if the landscape is constantly changing, like I've tried to show with this fitness treadmill, then the climber is going to proceed in the favored direction until it's blocked or limited in some way. Now, how could this effect work in the world of population genetics? Let's begin with the simplest possible case. Out of many possible possibilities, the climbing robot just faces two choices. Up and to the left or up and to the right. We can assign a mutation rate and a selection coefficient to each option. Now, obviously, if the option that has one option has both the higher selection coefficient and the higher mutation rate, that's the favored outcome. But the interesting case, the non-trivial case to consider, is where the option with the higher mutation rate isn't the one with the higher fitness. Now we can take this scenario with two possibilities and render it in a traditional form as a two-locus, two-allele model. That's the model of Jampolsky and Stolz, who's shown on the right of the slide. So starting with a little a, little b population, mutation can introduce little a, big B with the higher rate here, but the smaller selection coefficient, or it can introduce little a, big A, little b with the smaller mutation rate, but the higher selection coefficient. And setting this up as a two-locus, two-allele model creates this third option, which we don't want to consider, so we just give it a smaller selection coefficient. So we simulate evolution on this landscape using a variable mutation bias, and I'm going to show you some results where the selection coefficients are 0.01 and 0.02, so a twofold bias in selection coefficient. And we did this for a variety of population sizes, smaller ones where mutations are rare, and for larger populations that have a steady supply of new mutations. And this shows the bias in outcomes, going to the left versus going to the right, as a function of mutation bias. And the first thing to notice is that all the lines are going up. The behavior of smaller populations tends to converge on this gray dashed line, which represents origin-fixation dynamics. You can also think of this as strong selection, weak mutation dynamics. So origin-fixation dynamics emerge when the rate of introduction of new variants is so low that we can treat each one separately. Evolution is then depicted as a process of jumping from one state to the next, and the rate of this jumping process is simply the product of the rate of introduction and the probability of fixation. Under these conditions, a bias in mutation becomes a bias in the introduction process, which directly and proportionately biases evolution. So for instance, here we have the chance that the next step from the starting genotype for I will be of type J instead of type K. And if we render this as a ratio of two origin fixation probabilities, then we get a ratio of mutation rates and a ratio of probabilities of fixation. If we're talking about a case where we can use the approximation for beneficial fixations, the chance of fixation under a range of conditions is approximately 2s, and if we use that simplification, we get a ratio of mutation rates times a ratio of selection coefficients. And that's that dashed gray line here. So given that the total mutation rate is about 10 to the minus fifth, we expect the smaller populations here to be in the origin fixation regime or close to the origin fixation regime, and this is what we see. Now, let's contrast this behavior with the mutation pressure theory of Haldane and Fisher, which suggests that mutation will only be important if selection is absent or if mutation rates are abnormally high. Does this behavior that we're seeing arise because selection is weak? Does it arise because mutation rates are unusually high? Well, the way to answer this would be to change the magnitude of selection coefficients. So we can keep the ratio of selection coefficients that's the same, but change them from small to large. And likewise, we can keep the ratio of mutation rates the same, but reduce the mutation rates until they're very small. So here are the results of doing that. On the left, we see the bias in outcomes over a 200-fold range in the strength of selection, and the bias in outcomes remains the same, but these are for 3 different degrees of mutation bias over here. On the right's the effect of mutation rate. So we had a mutation rate of about 10 to the minus fifth previously, and this shows the effect of reducing that 1, 2, 3, 4 orders of magnitude, and the result is just that the bias in outcomes converges on origin fixation expectations as mutation becomes vanishingly rare. So this contradicts the thinking that says that mutational effects in evolution require high mutation rates or weak selection. In fact, we're going to see the strongest effect when mutation is weak and selection is infinitely strong, so that beneficial mutations are immediately accepted. In that case, the very first beneficial allele to be proposed will get accepted, and the dynamics will be entirely determined by what's mutationally likely. Why does that seem counterintuitive? Well, think about it. It seems counterintuitive because we've been taught to think the wrong way. We've been taught to think about the system being pushed around in a field of allele frequencies by mass action forces, and that's not the way to understand evolution. The behavior that seems counterintuitive relative to the forces theory emerges from the introduction process, and we can demonstrate this by removing the introduction process and putting the alternative alleles into the initial population the way we would do in classical thinking. So that's what I've done here. The upper line is one of the lines that we've seen before for a population size of 10,000. The lower lines are when we put the alternative alleles into the initial population. So in all 3 cases, mutation is still happening. In all 3 cases, mutation is biased. But that doesn't make any difference down here, and the reason is because the alleles are already present, so mutation is just shifting the frequency of things that are already there, and because mutation rates are so small, that process is not important, and biases in that process are not important. That's the thinking that leads to the Haldane-Fisher argument. Mutation pressure is not important because mutation rates are small. The bias that we see in the upper series is a bias in the introduction process. This is important to understand. The mutation bias and introduction bias are two different things. Okay, you can have mutation bias without introduction process, without a bias in introduction, which is what we have down here. There's no introduction process because the alleles are already present, so there's no mutational bias in introduction. Mutation bias can happen without inducing an introduction bias, and introduction biases are possible without mutation biases. We'll find out in a moment. All right, so that's the basic theory. And when we published this 20 years ago, I honestly thought that this was revolutionary, and I kept waiting for the revolution to start, and I'm still waiting for the revolution to start. That's not the paper hasn't been ignored. It's quite highly cited, but there was zero interest from theoreticians until recently. In just the past few years, a small body of theoretical work has emerged that builds on this basic finding, and I'm going to review this work very briefly without fully explaining it. So, last year, Suarez and colleagues carried out a more rigorous and thorough analysis of a model that's very much like the original Yampolsky-Stoltzfus model, except it just stipulates that there are two options left and right without using the two locus, two allele formalism. And the authors present simulations and mathematical results over a wide range, and they also do a model with three arbitrary alternatives, A, B, and C, instead of just A, B. So this model and the original Yampolsky-Stoltzfus model are really models of one-step adaptation at one locus or site. But most of the interesting work that has come out recently is about what happens in the long term or what happens across many sites in a gene. So what's the effect of mutation bias in longer adaptive walks over a fitness landscape? I'm going to start with a study that I did in 2006 addressing the effect of GCAT bias on amino acid composition. So organisms with higher GC content, they tend to have proteins with more of the blue amino acids here, GARP, which have high GC codons, and they have fewer of the red amino acids here, FYMINK, that have AT-rich codons. So what's going to happen if we simulate evolution of a protein coding gene adaptively under conditions of a bias in mutation that favors GC or that favors AT? So I looked at this problem using an abstract kind of fitness landscape called an NK model, and I found a strong effect of mutation bias on amino acid composition. So what we're seeing here is the ratio of those two kinds of amino acids, red to blue, under different kinds of mutation biases, the strong bias in the red direction, in the AT direction, or in the blue direction, the GC direction here. This is a tenfold, threefold, no bias, threefold in the other direction, and tenfold in the other direction. And the difference, so this shows the increase in composition bias over the course of an adaptive walk. There's a bunch of different walks here, but and they don't all have the same length, but the way that they're combined is just by stretching them all to the average length. And these walks, these two panels differ in that this is a rougher landscape over here, so the walks are shorter. It's a smaller number of steps until the population becomes stuck on a local peak. And this interesting recurving behavior here actually arises from a very generic aspect of adaptive walks on finite fitness landscapes. So steps that are kinetically favored, whether that's due to a high mutation rate or to a high selection coefficient, they tend to happen earlier in the walk, and then when you get to the end of the walk, there's some steps left, and those are enriched for the ones that are slow and they didn't happen earlier. So that's with this very abstract fitness landscape. What about real fitness landscapes? We don't have real fitness landscapes of a large scale for proteins, but theoretical landscapes can be computed for RNA folds, and there are also using algorithms that are very realistic. And empirical fitness landscapes are available for transcription factor binding sites. So I like to talk about those two cases. So first, the RNA folds. A property of the mapping of RNA folds to sequence space is the mapping of sequences is drastically unequal. Some folds have many, many sequences and others have few. In this study this is a figure from a study by Schopper and Lewis, where the authors show that RNA folds that are more mutationally discoverable due to having more sequences are more likely to evolve, and they show that this is an effect of mutation. I'll say more about this at the end of the talk. In this figure, they're showing a case that's set up a little bit like the Yampolsky-Stoltzfus model, where we have a starting fold, and there's two alternative folds that are both more fit. But The fold that has the higher selection coefficient here, P2, actually has fewer sequences. It has fewer genotypes, and it's less mutationally accessible from this starting fold. And this fold actually tends to evolve more just because it's more mutationally accessible, even though it doesn't provide the highest fitness benefit. Transcription factor binding sites provide another kind of complex landscape. So it's possible to synthesize all 8 nucleotide sequences and measure them using a high throughput method for how well they bind to a transcription factor. From the sequences that show appreciable binding, we can construct an affinity landscape, and then we can treat that like a fitness landscape for the case where fitness is proportional to affinity. Cano and Payne, in a 2020 study, used data from 700 or 800 of these landscapes, and for each one they identified the largest connected network of sequences that actually bind, and they used this as a fitness landscape. So here on the left we're seeing a histogram showing the composition of transition and transversion steps in those landscapes. The black bars are the background distribution of all steps in the landscape, and those are tightly clustered around 0.5 as we expect because there's twice as many transversions as transitions. But if you only look at upward steps in these networks, the steps that would be part of an adaptive walk to a peak, those are shown in white here. The composition of those steps is more broadly distributed, and it's actually a little bit pushed in the direction of transversions, which tend to have a larger effect on binding. So this is for like 700 or 800 landscapes, but they fall into 48 categories for 48 different types of transcription factor domains, and that's what's shown here, those 48 different types. So these data here are the same as these data here, but organized into 48 different binding types, binding protein types. So the universe of possible adaptive walks on these landscapes versus these landscapes versus these landscapes, it's going to be biased substantially toward transitions or toward transversions. So how will this influence evolution? We might expect that adaptation will be more or less effective depending on whether the mutation bias of the evolving system matches the composition bias of the landscape. And indeed, that's what Cano and Payne found. So what we're seeing here is dots showing the chances of reaching the global peak for populations with different degrees of mutation bias. And this curve differs reaches a different maximum on each of these 5 plots, and each one of these 5 is a different bin of the transcription factor fitness landscapes, separated by the degree of transversion bias. So this is a set that has the highest transversion bias, shown by this dotted vertical line over here, and this is the set that has the highest transition bias, shown over here, and then these are intermediates between those. And in each case you can see that the chance of reaching the peak is maximized approximately when the population has a mutation bias that corresponds to the composition bias of the landscape. And this is a really, really interesting result, and it allows us to say some interesting things about how search algorithms work, and to respond to the idea that mutation biases are some kind of limit or constraint on the power of selection. That's just not true. That's not a correct or accurate description of what's going on here, because there's no free lunch. Okay, there's no mutation spectrum that provides the best search, of which the actual mutation spectrum is some compromise. It's not a compromise. Different mutation spectra will be good on different landscapes. And the ones that are good on one kind of landscape are not going to be as good on another kind of landscape. So finally, I'm going to briefly review a completely different study that addresses effective mutation bias over a very, very broad range of mutation rates. So this is from Gomez and colleagues in 2020. They explored an abstract model with 2 traits, trait A and trait B. And each trait is subject to beneficial mutations, but for trait A, the selection coefficients are bigger, and for trait B, the mutation rates are bigger. Which trait's going to dominate adaptation? Well, the authors found that which trait dominates adaptation is just a matter of what's the intrinsic rate of increase in fitness due to this degree of mutation and fitness effect, or this degree of mutation and fitness effect. So in order to show their results, what they did was they just plotted that function. And what they've shown here is a contour plot. So this is a little bit difficult to read, so just bear with me. We have 12 orders of magnitude of mutation rate here, 2.5 orders of magnitude of selection coefficient. And these are 3 different contour lines showing the same rate in different regimes. And if there were no effect of mutation, the lines would be vertical, and if there were no effect of selection, they'd be flat. Okay, so the more vertical they are, actually, let me do it this way. So the more vertical they are, that's the higher the slope, the more the effect of selection. So down here in the origin-fixation regime, okay, so we've got a population of 10 to the 9th, but mutation rates down here are lower or smaller than the inverse of that. So we're in or near the origin-fixation regime. There's a linear effect of mutation. There's a linear effect of selection coefficient. But then when we start to get into the concurrent mutations regime here, where there's multiple mutations competing, the effect of selection is stronger due to that competition. But then there's this weird thing that happens in this diffusive regime, which is very new in this paper, and it's not something I've ever thought about, where mutations are not just trickling in, they're pouring in. There's like a mass action flow of new mutations when you get into this diffusive mutation regime, and you again see some proportional effects. So what this model is telling us is that mutational effects are important across a very broad range of conditions, but they're acting in different ways in different regimes of population genetics. All right, so let's take this theoretical work and put it into a broader context. What does this theory tell us, and how does it relate to challenges in evolutionary biology? In general, this theoretical work gives us a warrant to draw an arrow, like this green arrow, an arrow of causation, connecting cause and effect. Tendencies in mutagenesis or varigenesis can induce tendencies of evolutionary change, and this causal linkage is mediated by biases in the introduction process. Now, we want to apply causal theories like this to explaining and predicting evolutionary patterns. What kinds of explanations and predictions are relevant? In general, this is where the theory lives, between the left and the right sides of this diagram. On the left, we have tendencies of mutagenesis or varigenesis and the mutational, developmental, and structural causes of those tendencies. And on the right side, we have tendencies of evolution that might cause interesting patterns, things that I like to think about: patterns of molecular evolution, novelties, self-organization, the sense of findability, effects that might be assigned to developmental constraints. So the theory tells us conditions under which we can propose a link between things on the left and things on the right. It doesn't mean that the theory automatically explains these things. It just falls in the right category of testable hypotheses to link the things on the left with the things on the right. So the way to think about this theory is the kind of connections it can make, and in particular, what it adds to our ability to understand evolution relative to other theories about the role of variation, including the historically important theories that I've listed here. So for each of these theories, we can ask whether it justifies an arrow of causation that might link things on the left with things on the right. For instance, we've already seen results showing long-term directional trends in adaptive walks due to mutation biases. And this would be considered impossible or paradoxical in classical thinking. It contradicts what Haldane and Fisher said. None of these other theories provide for that. Importantly, this theory allows us to address the idea of developmental biases. So I've been talking up to this time about mutational biases, but if we leverage the concept of a genotype-phenotype map, we can talk about developmental biases. I already showed the genetic code, which is the genotype-phenotype map relating codon genotypes to amino acid phenotypes. So if we start, for instance, with a codon for this phenotype, aspartate, there are 2 different mutations to this phenotype, glutamate, and only 1 to this phenotype, valine. Even though there's no bias at the mutational level, this mapping of genotypes to phenotypes is going to induce a 2-fold bias in the introduction of the glutamate phenotype, and it's going to have the same effect in this model as if it were a mutation bias. Remember I said earlier that we can have mutation bias without an introduction bias, and we can have an introduction bias without mutation bias. This is how. Now this example uses the genetic code as a genotype-phenotype map, but it relates to something that people in evo-devo have been saying for decades, and which is a general property of genotype-phenotype maps, which is that they induce asymmetries in the mutational accessibility of alternative phenotypes. So if we were to make a generic kind of developmental example of this, we might say that here's a phenotype P0 that, or here's an alternative phenotype P2 for which 4 different mutations result in this phenotype, whereas only 1 different mutation results in phenotype 1. So in classical genetics, these 4 different similar phenotypes arising at different loci would be called phenocopies. And even if all the mutation rates are the same, there's still going to be a 4-fold bias. So that will induce a bias in the introduction process that will have the same effect of a 4-fold mutation bias, even though it's a developmental bias. It's not a mutational bias. And more generally, any kind of non-trivial genotype-phenotype map is going to induce this kind of asymmetry and also another kind of asymmetry. So let me just tell you about that briefly. So there's a local accessibility that's a function of the proximity or the shared contour length of genotypes. So when we're talking about, this is a stylized map showing RNA sequences with 3 different folds, the gray, the green, and the blue. So each point is a genotype, and they're all connected by mutations, and they all have the same fold if they're the same color. So phenotype P2 is much more mutationally accessible from P0 than phenotype P1. Even though they both have the same number of genotypes, there's more shared contour length here. It's mutationally closer. And so because of this effect, if you start with P0, you're more likely to evolve to P2 than to P1. There's also a second effect that's important in long-term traverses of sequence space, which is an effect of the total size of a network. So here P0 has about twice as many genotypes as P2 and P1, and this makes it roughly twice as findable in long-term traverses of sequence space. So to put this in a different way, larger phenotypic networks have more mutational arrows pointed at them from other regions of sequence space, which makes them more findable by an evolutionary algorithm that searches sequence space through mutation. For many decades, there's been a body of literature arguing for the evolutionary importance of effects that kind of sound like this. So Kaufman brought attention to this type of argument in The Origins of Order in 1993. And in the literature, effects of findability of common structures are typically described in terms of background conditions, as properties of the landscape, or in terms of self-organization, rather than being described by pointing to a particular population genetic cause. However, we've been arguing for a long time that the theory of biases in the introduction process provides the proper causal foundation for understanding these claims. And work from Ard Lewis's laboratory that I just cited in the past 5 years has confirmed this, has really nailed this down, that findability is a consequence of kinetic bias due to mutational introduction, and it's an effect that they call the arrival of the frequent, or arrival bias. Okay, thanks for listening. What we've learned in this lecture is that there is a body of formal theory that gives us a warrant to draw an arrow of causation between tendencies of varigenesis or mutagenesis and tendencies of evolution. And this is significant not just because this kind of arrow was previously understood to be incompatible with population genetics, but also because it's useful. It could help us to understand a variety of claims about things like developmental bias or findability. And more generally, it provides a basis for us to reconsider internalist ways of thinking that never really completely disappeared from evolutionary biology, but which have been kind of waging a struggle for legitimacy ever since the modern synthesis established a kind of default neo-Darwinian view and made all the alternatives seem crazy. So for instance, we can now specify a clear causal grounding to consider the idea of trends, directional trends caused by internal variational biases, which is something that Simpson and the architects of the modern synthesis dismissed as an appeal to mystical inner urges. Well, it's not due to mystical inner urges, or at least not necessarily. We can specify a population genetic grounding for this. But just remember, because of this history in which internalist ideas have been ridiculed and dismissed for being mystical or for being metaphorical, it's important to always keep in mind that this line of argument refers to a specific theory. This is not a mashup of old ideas about chance or constraints. This is not a case of using new words for old concepts. Instead, there is a specific kind of causal behavior that emerges when we put our focus on the transient of an allele frequency as it departs from zero and becomes non-zero. That is where the action happens. Focusing on this aspect of evolutionary dynamics leads to a specific theory that's not like other theories. We'll go into this in more detail in the next lecture in this theory, in this series, when I contrast this theory with other theories like mutation pressure, raw materials, quantitative genetics, and so on. So thanks again for listening. I'm now going to scroll silently through the final bibliography slides for those who are interested in exploring sources.